is this?
I'm Nemo.
Well, Nemo, all new explorers must answer a science question.
Okay.
You live in what kind of home? Okay, don't hurt yourself. Welcome aboard, Explorer!
Hey there, my name is Wes, and I'll be talking to you about the av3 sea anemone neurotoxin, which ion channel this toxin modulates and how. We're going to look at the structure of the toxin, what it's used for, and how it's used. What is an av3 sea anemone neurotoxin, and what does it do? It is a neurotoxin from the sea anemone viridian, and it binds only to the arthropod voltage-gated sodium channels. It is inactive on mammals. Experimental results suggest that this is due to a different sequence in the channel. Mammals have tyrosine located at a linker, where arthropods have histidine. It inhibits the inactivation process of voltage-gated sodium channels that bind to receptor site 3. Inactivation of the sodium ion channels causes sodium ion flow through the channel to stop. As a consequence, the membrane potential will stop rising. With the sodium no longer contributing to the membrane potential, the potential decreases back to its resting potential as the neuron repolarizes and then hyperpolarizes. Here we have an image of the bioactive surface of the av3 neurotoxin. The image shows both sides of the structure, so we can see all the residues it is made up of. It is a 27 amino acid long residue, and as shown in the image, the residues are color coordinated. Tyrosine, phenylalanine, tryptophan are in magenta, proline and asparagine are colored in green, arginine and lysine are colored in blue. How does a neurotoxin act on the sodium ion channel? Voltage-gated sodium channels respond to changes in membrane potential. This change in membrane potential opens the channel pore, enabling sodium ion conductance. Voltage-gated sodium ion channels are targeted by many venomous toxins used by organisms such as sea anemones and scorpions, for hunting and in defense. Sea anemones depend heavily on their venom for survival. They paralyze or scare off their prey or foe by stinging them with a neurotoxin. There is a video later in the presentation which shows how they sting their foes. Structure of the voltage-gated sodium channel. Voltage-gated sodium channels of most animals are large membrane proteins made up of a pore-forming alpha subunit and one or two auxiliary subunits. The alpha subunit is made up of 4 domains, each of which contains 6 transmembrane segments, all connected by intra- and extracellular linkers. Receptor site 3 is located at the linkers S5 to S6 at DI, S1 to S2, and S3 to S4 at DIV. This can be seen in the image. Just a quick summary on how the toxin is used. Sea anemones depend heavily on their venom for survival, as previously stated. They paralyze or scare off their foe by stinging them with a neurotoxin. As the foe is stung with the av3 neurotoxin, the neurotoxin binds to the voltage-gated sodium channel and inactivates it. This causes pain to the organism and as a result protects the sea anemone from any harm.
Nematocysts are stinging cells common to all anemones and jellyfish. When stimulated, they fire a venomous dart attached to a thread into their prey.
We can look at the use of the tentacles to trap food by taking a small piece of food, this is a little piece of limpet, and dropping it over the ring of tentacles. They kind of close in on it and pull it down.
Oh, it likes that. It likes that a lot. It's
got a very hungry anemone here. And it's closing all of the tentacles, all 6 rings. It's closing them in to push the food back down towards its mouth. And they're not just for trapping prey either.